All right, so hello everyone. Today's lecture will be about combining neuroscience methods. My name is Mads Wisniewski. I work at the University of Minnesota as a postdoctoral researcher, and to be specific, at the Department of Biomedical Engineering. As I said, today we will be talking about combining neuroscience methods. So let's quickly look at an overview. First of all, I will be talking about why we want to combine neuroscience methods, why that would be a good idea to combine them at the same time rather than just sequentially, what the advantages are, and what the problems with that are. And then I will go through some various examples of combined neuroscience methods. First, I will be talking about fMRI and EEG, then about fNIRS and EEG, TMS, EEG, and finally about TMS, fMRI. And then at the end, I quickly want to talk a little bit about closed loop systems, which is a relatively novel technology that I think is really cool. And I say that because I'm working with that. And as you can see here in this overview, combining different methods with each other combines different dimensions with the other. So for example, this temporal and spatial dimension, or the correlational versus interference dimension. So let's start with defining what that means. So this picture is one that you may recognize because you will see it in most neuroscience books, or at least a version of this figure. And this figure shows on 3 different axes where each of the neuroscience methods has its strengths and weaknesses. And that could be either at the temporal, spatial dimension, or the correlational and interference dimension. So let's talk about the spatial dimension. Some methods are spatially very precise, which means that you can look at a very small part of the brain. If your spatial resolution is very, very high, you would be able to look at single neurons or even just fractions of a single neuron, for example, just the dendrites or just the axon, or maybe just the synapse. However, that's typically not possible in alive humans. To be able to look at a single neuron, you would have to do something like single cell recording, which means that you would have to plant an electrode in the brain, and in alive humans that's typically impossible because you would have to operate to get that electrode in there. So spatial resolution in humans overall will always be a little bit limited. However, some methods are still better than others. And of course, you would want to combine those, the ones that are very good spatially, with ones that are temporally very precise. So what do I mean with that? Some methods, they can measure the brain activity at the millisecond that it happens, which means that temporal resolution is very good, whereas other methods, they are temporally not very precise, which means that the activity that they're measuring is not very precise in time. There could be a delay, or it's just quite variable. So combining a good temporal with a good spatial resolution would of course be the best thing out there. And then there's dimension correlational versus interference. Correlational means that you're just measuring activity, but you're not changing the activity, which means if you're just looking at a correlational measure, you cannot say that a particular region or a particular pattern in the brain activity is necessary for a particular function. You can just see that it is active during a certain task, but you're not sure if it's necessary. To be able to say something about this necessity or making a causal conclusion, you would have to interfere with an activity. So you have to change the activity to know whether a particular region is really important for a specific task. Now, there are some other dimensions that are not shown in this figure that I quickly just want to mention. There is the invasive versus non-invasive dimension. invasive means that you actually hurt the brain or damage the brain. And that's typically not done in humans. It's not something we would do. So in most methods in humans are non-invasive methods, which means that you do not damage the brain. So we don't need to really talk about that particular dimension today. The last dimension that I want to mention is the brain versus behavior dimension. Because of course in neuroscience we want to couple brain activity to a certain behavior in humans. So combining a neuroscience method with behavior, that's something that is typically done quite a lot. You give a certain task and observe what the brain activity is. But there are some intermediate things there that I quickly want to talk about. So what do I mean with this dimension? So you have, of course, behavioral tasks, let's say a memory task or an attention task, and you're measuring a certain outcome measure, reaction time, accuracy, you name it. And you can combine that with an imaging method or an interference method like MRI, EEG, or TMS. And that's not something special. You can do those two things at the same time. However, there are some things you can record that are a little bit intermediate. Two examples that I want to give is electromyography and eye tracking. So eye tracking, for example, allows you to look at the eye movements, for example, during a task, a spatial task. When you see different stimuli appearing on the screen, you can see where the eye goes towards. And since the information that enters the eye is directly going to the visual cortex, that is already somewhat an indirect measurement of brain activity. You're not measuring the brain directly, but you're also not just looking at the behavior, right? You're not just looking at eye movements, but that those will tell you a little bit about how this will translate to brain activity. And the same is true for electromyography. By measuring activity in a muscle, you kind of get an indirect measure of what's going on in the motor cortex, because of course the motor cortex is directly coupled to the muscle output. So these two methods are some intermediate methods that you can combine either with a task or with the imaging/interference methods, MRI, EEG, and TMS, to get more information about some physiological things going on. But to be fair, combining those with those other methods is relatively easy. It's relatively straightforward. You don't need to take anything into consideration. You just have to have the machine and measure it. So I won't be talking about it more in this lecture. I really want to focus on combining those three methods here: MRI, EEG, and TMS. So first, look at combining spatial and temporal dimensions to get both a very high spatial resolution and a very high temporal resolution. And the first thing that would pop up in your mind would be fMRI and EEG. So first, let's have a look at the advantages and disadvantages of both methods. The advantage of fMRI is that it's spatially quite specific, where the spatial specificity of fMRI will depend a little bit on the magnetic field strength that your MRI machine has, but a 3 Tesla scanner typically has a spatial resolution of 3 cubic millimeters, 2 to 3 cubic millimeters, which is, relatively small. It's not single neuron level, but it is, in humans using a non-invasive method, the best thing we have. However, the time resolution is not very good. Why? Because with fMRI we measure the oxygenation of the blood, and so how the oxygen goes to the neurons after they have been active. So what we see is a neuron becomes active, or a group of neurons become active, and then after a while oxygen is transferred to that area. However, this is a physiological response of the body that takes time. It takes between 6 to 10 seconds, and it's quite variable, that time, which means you are limited in how specific you can be time-wise. There will always be at least a 6-second delay, but it could also be an 8-second delay, and you just simply don't know. Which means that your temporal precision is somewhere between 2 to 3 to 4 seconds at best, which is not very nice if you're giving a task where different processes in the brain happen within a few hundreds of milliseconds. And yeah, so that's why you would want to combine this with a method that is temporally very precise, such as EEG, because the advantage of EEG is that you're measuring electrical signals on the scalp that come from the brain, and you can measure them basically at a millisecond precision. So when it happens in the brain, you will also see it. So the timing specificity is in the millisecond range, and that's very, very good. However, of course, the disadvantage of EEG is that it's spatially not very, very good. When you see activity on the scalp with EEG, you have a little bit of a problem because you don't know where that activity comes from, and that's what we call the inverse problem. What is that? So when you measure a certain pattern on the scalp with the EEG electrodes, you cannot really say what the sources of that are within the brain. For example, two sources, so 2 points of activity, could lead to the same output on the scalp as just a single point of activity. And since you don't know how many active hotspots there are in the brain, you cannot really resolve where the activity comes from by just looking on the outside. All right, so combining those two methods would be a good idea. EEG is temporally very specific, fMRI is spatially quite specific. So put on an EEG cap, put them in an fMRI machine, measure EEG activities, just like here in this slide, for example, with oscillations. Look at where this activity is in the MRI, and voila, you got it. Unfortunately, it's not that easy. Because first of all, you need to have specific materials. You cannot simply put any EEG system in the MRI machine. Why not? Because it's not MR safe, because it has magnetic materials, like metal, for example. So you need to have an EEG system that only has non-magnetic materials. So that includes the EEG electrodes. Of course, those small electrodes, they often in a normal EEG system are metallic, and you would need something that is non-magnetic. And of course also the amplifier and the battery of the EEG system, which also would have to be near the MRI scanner, need to be non-magnetic. And also the gel that you use should be safe. Now those things are commercially available. So if you have the money, you can buy this and you will be fine. You just have to be aware of it. However, there is another problem that cannot be solved by having money, and that's the fact that you will measure artifacts. So what type of artifacts can you expect from combining the two methods? Well, you will see artifacts both in your fMRI signal as well as in the EEG signal. So in the fMRI signal, if you have ever been into an MRI machine, the experimenter probably would have asked you to not cross your legs or cross your arms, because this creates a loop. And creating a loop means that basically an electric current goes through that loop, because of course you are in a big magnet, and via induction magnetic field will cause an electrical field, and so in a loop that will create like an electric current, and that causes a disruption of the signal. Now, of course, the EEG electrodes, which are round, basically are just a lot of small loops. So that will cause a distortion of the MRI signal. And also the gel itself can cause a distortion because there's a lot of fluid in gel, and the MRI signal is dependent on hydrogen, so water basically. So yeah, that the specific gel you're using and how much gel you're using can also distort the MRI signal. The biggest problem though, which you will probably find in the EEG signal, as you can see here on the left. On the left you will see a signal of EEG that looks perfectly fine, and then if you put it into the MRI signal, it gets completely distorted. It gets distorted by the MRI field itself, so the gradient of the magnetic field, but also by the radio frequency pulse that is used to actually measure the magnetization, and also the ballistocardiographic artifact, which is basically an artifact induced by your heart rhythm, basically. Now you can get rid of those artifacts. The radio frequency pulse can be removed by an ICA component analysis. A magnetic field distortion can be disrupted by using different kinds of filters and also an ICA component analysis. And the ballistocardiogram can be controlled for by actually measuring heart rate in the MRI scanner. However, always keep in mind that when you have a distorted signal and you're trying to correct for those distortions, if you try to correct for those artifacts, the signal that you will be left with is always worse than a signal that has no distortions at all, because of course you're basically manipulating your signal, and you will always you will get rid of the artifact, but you will always eliminate some real signal, basically some actual brain signal. So your signal to noise ratio will be a lot worse than if you would not combine those methods. So always keep in mind that an EEG signal, or an fMRI signal in that case, in a combined method is always suboptimal, which is always something you need to take into account when looking at those signals. However, of course, the advantage, of course, of combining methods was the spatial and temporal resolution. So it really, it has advantages and disadvantages. And if you do it correctly, you can get actually quite nice findings. For example, in this study by Ragazzoni et al., they have done something really cool. In the MRI scanner, they recorded EEG and they gave people an oddball task. And an oddball task is basically a task where you get to see a lot of the same stimuli and all of a sudden something changes. And that change will cause an attentional component because, hey, something changed. What was that? That's basically the signal you get. And you can get this attentional oddball response by either actually presenting a stimulus that is an oddball, so a stimulus that is different from all the other stimuli, or by just omitting a stimulus. So you basically, if you would give a sound, you would have beep, 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 and then beep. A higher sound, that would be an oddball, or just omitting the sound that completely would be the omitted target. Now, when you just measure EEG during these kind of oddball tasks, you will see that there are components around 300 milliseconds, 200 to 300 milliseconds, that you can see all over the brain that are increased in frontal, central, and parietal electrodes. And the signal is called a P3. It has also some different names like the PP2, but it's typically referred to as the P3 component. And we know that this basically is this attentional component. You get the surprise component, basically. And that has been known for decades in EEG research. The problem, however, is because you measure it all over the brain with EEG, or all over the head with EEG, for a long time it wasn't really known what the source of this signal was, which is problematic, and it couldn't be solved with just EEG. So what this study is, they combine it to see where that signal comes from. And what they found is that actually an oddball, so like a different stimulus, evokes some activity in the anterior interparietal sulcus, whereas an omitted target has increased activity in the medial frontal gyrus and the cingulate cortex. And that's already interesting because based on the EEG studies that have been done before, it wasn't really clear that an oddball or an omitted target would be related to different areas in the brain, because both the oddball or an omitted target is a surprise response, and they both show a P3 kind of signal. So it was quite surprising to see that those are related to different areas in the brain, or at least to unique areas in the brain, because there is also some overlap, the inferior frontal gyrus and the supplementary motor area. So, and that is of course what was mostly expected. Yeah, so this is a nice example of a study where they combined the two methods to find specific locations in the brain related on a very timely specific response around 300 milliseconds after a stimulus is shown, or in this case not shown. Another way to combine the blood oxygenation level dependent response with EEG is with fNIRS. So fNIRS is another method to basically measure the same thing as an fMRI machine does, the oxygenation of the blood that goes to the neurons to provide oxygen. The advantages of fNIRS are clear. You can see it on the picture. It's a lot smaller. The electrodes are a lot smaller. You're not in a huge MRI machine. So how does it work? It basically sends infrared signals through the skull, and those loop back to the outside and are measured by other electrodes. So the red ones, I think, send the infrared signal in the brain, and then they come out in the white electrodes. And then you basically measure the same thing as with fMRI. So that's really cool. And you can combine it with EEG. There are actually, like this cap, there are commercially available combined caps. So that's really cool. So the advantages of fNIRS are clear. It's a lot easier to integrate with EEG. There are definitely way less problems with artifacts because you don't have a huge magnetic field. And it's also a lot cheaper than fMRI. So basically, why not go with fNIRS? Well, it has quite a big disadvantage, and that is that the spatial resolution is a lot worse than fMRI, and also it cannot measure as deep as fMRI. fMRI can basically look at the entire brain, whereas fNIRS can only look at the surface. And the spatial resolution of fNIRS is around 2 cubic centimeters, which is 10 times worse than fMRI. And since the combination with EEG would be to combine spatial and temporal resolution, having not a great spatial resolution kind of eliminates the point of combining those two methods. fNIRS is still spatially more specific than EEG is, but the advantages of combining those two methods are not as great. And of course, fNIRS, because it depends on this oxygenation, this physiological response of the body in the brain, its temporal resolution is also as bad as an fMRI. But that's why we would want to combine it with EEG. So, but yeah, since not so spatially ideal, it's not that used that often, unfortunately. However, there are some cool examples of it. For example, this study, they identified an area that is related to arithmetic ability. So they basically measured fNIRS and EEG at the same time, and they measured this in people with a high mathematical ability and a low mathematical ability, and they found that there was a difference in activity in the left inferior frontal gyrus. Now note that this area looks very specific, but this is just the difference between those two. If you would look overall, then those areas would not be as precise as you would see here. And then they looked at the EEG signal that was measured at the same time while the people were doing a subtraction task, and they basically coupled this left inferior frontal gyrus response to a EEG signal around 200 to 400 milliseconds after people had to do this or saw the stimulus, the arithmetic stimulus. Now you could wonder, this could easily have been done, well, not easily, but it could have been done with fMRI EEG, and then probably your spatial resolution would have been a lot better. Why not combine it then with fMRI? Well, this particular example, this particular study was based on a previous study of the same group where they did it in children. And as you may know, in an MRI machine, you have to stay very, very still for up to an hour or even more. And, keeping a child still for that amount of time can be very problematic. The same would be true with people who may have a disorder that leads to tremors or something like that. So you cannot measure anything in an MRI machine in these populations. And in that case, the fNIRS method, although it's spatially less specific, has the advantage that you can actually measure something, right? So it's not ideal, but it's still better than nothing. So that's why fNIRS and EEG combined still has its advantages and still has its uses. Although, as I said, it's probably not used as much as fMRI EEG. All right, so let's have a look at combining correlational and interfering dimensions. As I said, correlational dimension looks at the activity but doesn't know if something is causally involved with a certain task. And the one interfering method that is well known and well used in humans is transcranial magnetic stimulation, whereby a short magnetic pulse is used to either interfere with brain activity or activate a certain brain area that you target. So what are the so again, let's first look at the TMS itself. What are the advantages of TMS? So it's an interference method, so it can activate or disrupt cortical activity, and that will give you some idea about whether a region is causally involved with a certain task. And spatially, it's more or less focal. It's quite okay. It has a resolution of around 1 square centimeter, though 1 cubic centimeter. Although it should be mentioned, of course, that if you activate something, that in turn will activate other regions, and basically you're hitting an entire network. So there are some people say it's not that spatially specific because you're targeting an entire network. But still, it's more specific than EEG is. But of course, the clear disadvantages is that you have a coil on your head and you do something to the brain, but you cannot actually measuring what you're actually changing. You have to you can see it in the behavior, but you cannot, at least not with TMS alone, measure any change. So that's where combining TMS with EEG would be a great method. So what often is done with TMS to actually measure its effect on physiology is that if you apply TMS over the motor cortex, we can measure the muscle output by electromyography. So you're basically placing the coil of the motor cortex, you activate the motor cortex, and that in turn will activate the muscle, and then you can measure the muscle activity. And that's actually quite a good output measure of what TMS does to the brain. However, that depends on having a muscle, and you can only do this for the motor cortex. For any other regions in the brain, frontal cortex, parietal cortex, auditory cortex, you name it, you're basically blind. You cannot use the EMG method. Of course, EEG can measure all parts of the brain. So if you could combine TMS with EEG, that would solve that problem. And that's basically what we're trying to do. Combining TMS with EEG to know what TMS actually interferes with and to see what the TMS actually activates. But of course, as you would expect, given that TMS is also just a magnetic pulse, there will be some artifacts and there will also be some confounding effects that I want to quickly talk about. So when you apply a TMS pulse while actually measuring EEG, it will more or less look a little bit like this, the signal here. You will first see a huge disruption of the EEG signal, which is basically the artifact when the pulse is given. And because that signal is so strong, it will saturate the EEG electrodes, EEG amplifier, which means that what you can see here is that it sometimes takes a little bit of time for the EEG signal to respond to its normal level, basically. And when the circumstances are ideal, that artifact combined with that recovery of the signal will take about 20 milliseconds. You cannot be, like, the artifact cannot be made much smaller than this. However, that's in ideal circumstances, which means that you have a TMS-EEG compatible system. Again, you need EEG electrodes that are not disrupted too much by a magnetic field. You need a very high sampling rate, and you need a very good signal-to-noise ratio. So your EEG signal needs to be perfect before you actually start doing the TMS-EEG. And also there are some other things you would have to look out for. For example, you have to orient all the wires of the EEG electrodes in the same direction. So these are all small things that you would have to take into account. And then if you're really good at it and if you've practiced a lot, you can get this artifact down to 20 milliseconds, which means that, you know, there is some loss of temporal specificity because the first 20 milliseconds are gone. Then again, that's still quite good. All right, and then what you will see is that this will, depending on the region where you stimulate, invoke some responses in the EEG signal, and these are called TMS evoked potentials. This is an example of when it's done over the motor cortex. You can do this over other regions of the brain, and it will look slightly different. But yeah, there are some. It will be quite consistent when you stay in one region. And over the years, a lot of research has been done by identifying what those different components mean. For example, this N1 response, which is seen over various areas, is most likely related to inhibitory GABAergic neuron activity. So inhibitory neurons in the particular cortex that you're activating. So if you have a region that you activate with more inhibitory GABAergic neurons, that response might be bigger as well. Whereas the P2 component is more related to glutamatergic activating neurons. So for example, if you would give a certain intervention, maybe a drug, a pharmaceutical drug, to increase glutamatergic activity in the brain, then you could measure this by applying TMS-EEG, and then you would see an increase in the P2 component. So that's really cool. You can see with combining those two methods, the change in activation induced by the TMS, and that tells you a little bit about how active that brain area is, but not in a correlational sense, but in a causal sense. So this area is more active because things changed, because you interfered with it. However, when interpreting the signal, you have to be a little bit careful, because there will, when you give a TMS pulse, if you ever had TMS before, you will notice, you will feel that on the head. It will give you like a tap, and also you will hear it. You hear a clicking sound, and of course, when you hear a clicking sound, that will evoke a sensory response. You will hear it, the auditory response, and the tap on the head you will feel, which gives you so much a sensory response. And those responses will also happen in that same time frame. And depending on where you're measuring, you will also pick this up with EEG, because as I said before, EEG is spatially not very specific, so that will pick up things that are happening all over the brain. So what you need to do is when you want to interpret the signal in a good way, you have to not only apply TMS-EEG, but you also have to use a sham version of TMS. So a sham version is basically a placebo version where you only let people hear the sound of TMS without actually using TMS, or just the tap on the head. The sound can be imitated quite well. The tap on the head is hard to imitate. There are not very good controls for that right now, so that's a little bit tricky to get rid of. So, and when you measure these control signals, you can subtract them from this TMS-EEG signal, and then you will have basically the true TMS-EEG signal. You can also look at the oscillatory response, and that is quite interesting because this is a study of Toot et al. From 2011, where they applied 5 pulses in a row, and there was always 10 milliseconds between each pulse. 100 milliseconds between each pulse, which means it's a frequency of 10 Hertz. So 5 pulses in a frequency of 10 Hertz. And what you saw is that the alpha activity in the brain was increased after those 5 pulses for up to 5 seconds after those 5 pulses occurred. So those 5 pulses occur within 500 milliseconds, right? Because there's one for each 100 milliseconds. And then for 5 seconds onwards, you will see a change in alpha activity, which also shows that repeated TMS pulses can have a prolonged effect. Of course, 5 seconds is not very long, but 5 pulses are also not that many. So if you would give more pulses, then the effect may be a lot longer. Here's, by the way, also an example of where they controlled for auditory and sensory things, and this effect was still present. Here's another example, by the way, where they done basically the same thing, but then in the visual cortex. Here the effect only lasted for a second, but still, that's quite good. All right, so let's see an example of why this kind of research would be important. And this is a study by Hill et al. From a group in Australia, where they basically looked at TMS oscillations in depression. And what they found is that compared to healthy control participants, the activity of lower frequency EEG waves in the delta, theta, and alpha range was increased in participants with major depressive disorder compared to healthy controls. And they measured this by applying a TMS pulse over the dorsolateral prefrontal cortex, which is based on a lot of research probably involved with depressive symptoms. So without going into too much detail about the study, because this was quite an elaborate study, but it shows how we can find causal markers of, for example, a disease such as depression. And so we know that this, because we interfered with this method with TMS, we know that these delta, theta, and alpha oscillations are causally involved with the symptoms of depression compared to healthy controls. And that's quite cool, because then we know what we need to target if we want to treat depressive symptoms. All right. So of course you can also combine TMS and fMRI, and when you do that, I would advise you to not do it like in this picture here, because this is not going to work at all. Because in actuality it looks quite a little bit different. A participant has to lay down in a specific manner, and not typically, like laying down in an MRI machine is already not very comfortable. But when you get TMS at the same time, it's even less comfortable, because the TMS coil can only be positioned in specific ways, right? You cannot, because you're limited in how the participant is lying down, and the MRI machine is also, the space you have is very limited. You can only orient the coil in specific ways, for example, here to the side or here at the top of the head, which means that the participant also has to lie down in a specific way to actually hit the target. And another problem is not only the space, but also that you cannot use any orientation of the coil that you want, because you're basically applying a magnetic field with TMS, and you're already in a magnetic field of the MRI scanner, which means if you use in specific orientations, those two magnetic fields will sum up, and that can lead to quite heavy magnetic pulses that you don't want to have in an MRI scanner. That can be a little bit problematic, and would completely disrupt the signal. I mean, So again, just as with with EEG fMRI, you need MR-safe materials, which means the TMS coil is non-magnetic, and also the cables and the machinery of the TMS coil. They are typically quite big, particularly the thing that produces the pulses. They are typically quite big and wouldn't fit near an MRI machine, and it's also hard to make them completely non-magnetic. So what you typically have to do is you have to use a very long TMS cable, which basically is so long that you can use the machinery outside of the MRI room. And of course that will also depend on how big the MRI room is in your specific lab. If you have a very big fMRI room, then your cable needs to be extremely long, and it may not even be possible to combine TMS with fMRI. We talked about this before, right? That the coil locations are very limited, and also the participant cannot move at all. Of course, in an MRI machine, participants shouldn't move in the first place. So, but even microscopic movements will, of course, cause the TMS to target a different area, and then your entire research question is out of the window. And yeah, what we talked before as well is that there will be artifacts, right? A magnetic field within another magnetic field that will definitely cause some artifacts. But again, if you combine it like in the best way possible, then you can have some quite interesting findings. And this study is a cool example of this, because they stimulated with TMS coil over the left dorsolateral prefrontal cortex, which is basically this area here, but then on the other side. And then you will see that this is a cut out part of the brain, so this is the inside of the brain. And what they saw is that stimulation of TMS over the dorsolateral prefrontal cortex also caused some activity compared to a control condition in the anterior cingulate cortex and in the caudate nucleus, which are very deep in the brain. Particularly the caudate nucleus is very deep in the brain and is at least multiple centimeters away from where the actual target TMS is. And that also shows, like I said before, that if you stimulate with TMS, you will hit an entire network. And basically what they did here is that if you stimulate the DLPFC, what network are you targeting? And in their case, they found that you target a network of DLPFC, anterior cingulate cortex, and caudate nucleus. So that tells you a little bit about the interactions, the causal interactions of the brain, the functional, how the areas are functionally coupled in the brain. All right. So last part, I want to talk quickly about closed loop systems, because what we talked about now before are open loop systems, which basically means you are applying TMS and you're applying EEG or fMRI, but you're not you're measuring them at the same time, but they don't influence each other. It's not like you change your TMS based on the signal you're getting, and that's kind of what closed loop TMS does, and a method that has been developed just a few years ago, and that we have developed as well in our lab, is a closed loop TMS-EEG system, where you read out the ongoing EEG activity. So basically your participant is sitting there and you are measuring EEG, and then while you're doing that, you give a TMS pulse, but that TMS pulse will depend on how the EEG signal looks like. So you're basically measuring the EEG, and based on what you're measuring, you are applying a TMS pulse. So that means you do it online. It happens while you're looking at it. It's super timing specific, and you can target specific parts of a EEG wave. So for example, if you want to target just the top of the wave or just the bottom of the wave, you can do that. So it can be phase specific and frequency specific. And that is a very unique and new way that has not been done before. However, that's very challenging, as you can imagine, because you have to measure EEG, but also you have to pre-process and analyze the EEG data, while at the same time also triggering the TMS machine. So the pre-processing and analysis and the triggering of the TMS machine all needs to happen within a few seconds. Within a few milliseconds, not in a few seconds, within a few milliseconds. You want to be timing specific. So in the method that we have developed is that we are looking at the signal, we are extracting that signal, and based on how the signal looks like in a particular participant at a particular time, we make a forward prediction, which basically means that we predict how the signal will look like based on the previous signal, so that we have a few milliseconds in advance that we know, okay, if we now stimulate in, let's say, 2 to 3 milliseconds, then we will hit this part of the EEG wave. So the difference between the green part and the red part here is about 2 to 3 milliseconds. So that's really cool, and in that way you are able to hit specific phases. And we have done it, and actually it's an ongoing study, and these are just some preliminary data, so this is not published yet. But we were able to actually hit the different phases of the alpha activity, or the mu rhythm in the motor cortex. The mu rhythm is basically the alpha activity related to the motor cortex, and that is important for inducing a movement. So we stimulated at the peak, at the falling phase, so is when the oscillation goes down, at the trough, and at the rising phase when the signal goes up again. And we measured the motor evoked potentials, so the output to the muscle that the TMS induces. So we not only combined TMS with EEG in a closed loop way, but we also measured the EMG at the same time. Those polar plots, by the way, show that we were actually quite successful at hitting the different phases. So it's not just that we have to trust that our algorithm actually works. We were actually able to see that it actually works. And as I said, these are just some preliminary data of a single participant. So this is not conclusive at all. But what we did find is that the output to the muscle is a little bit bigger when you stimulate at the peak phase compared to the other phases. And that's quite interesting, because if that, this is just a single participant, but if that would be true for more participants, then you could use that information if you want to use TMS to actually activate a certain region, right? So if you want to optimally activate a certain region, you would be better off by stimulating only the peak phase of a certain oscillation. And if that, as I said, this research still needs to be done, and maybe it's not the peak but the trough, but anyway, it would give you some information about a particular participant at a particular time, what the optimal way is to stimulate, and that and since TMS is used for treatment of depression and other disorders, you could optimize those treatments with that technique, and that's really cool. All right, so with that, I want to say thank you for your attention. I hope you enjoyed, and yeah, let me know what you think.